Good afternoon and good morning for those who are online and out west, and welcome to what I'm sure is going to be an informative and exciting workshop or symposium titled High Density and High Resolution Neurophysiology to Reveal Local Microcircuits in the Human Brain. I believe this may be the symposium title that incorporates the most brain initiative themes, so we're already off to a good start. I'm Lee Hochberg, Director of the BrainGate Clinical Trials and Consortium, and I'm a neurologist and neuroscientist at Massachusetts General Hospital, Brown University, and the VA. It is my privilege to be invited to moderate today's symposium. I'd like to thank the Brain Initiative Program Committee, Dr. Samir Sheff, and not only for this opportunity, but really for putting together such a tremendous meeting. One of the longstanding goals of the Brain Initiative, as everybody in here knows, has been to develop novel tools to better understand human brain function and to use our advanced understanding of neurophysiology to improve the diagnosis and the treatment of neurologic disease and injury toward restoring lost neurologic function. Having spent the last week attending in a neurointensive care unit, I can attest to the urgency of further developing a circuit-based and systems-based understanding of the human brain, and to using tools to bring that understanding to do carefully crafted research together in partnership with our patients and our research participants. Today's speakers are engaged in some of the most exciting research at the intersection of neuroscience and neuroengineering and clinical medicine, and I am really looking forward to learning from them. So, I'm going to leave as much time for the science as possible, so let's get right to it. We're going to have 4 speakers today, about 15 minutes each. We'll then have a 20-minute Q&A or panel discussion, and then we will wrap. So first up, it is my pleasure to introduce Dr. Angelique Paulk, who received her undergraduate and master's degree at Cornell, her PhD at the University of Arizona, and was a postdoc at the Queensland Brain Institute in Australia, all using a variety of electrophysiologic tools to study brain activity in bees and flies. She's currently an instructor of neurology at Mass General Hospital and Harvard Medical School, carrying out research in neurophysiology, neuroscience, and behavior focused on understanding the neural underpinnings of behavior leading to treatments in neurological and neuropsychiatric disease. At Mass General, Angelique works to study both neural and cognitive processing in the human brain as recorded using intracranial electrodes, including investigating the mechanisms of how direct electrical stimulation alters processing and behavior and translating novel brain recordings into clinical environments. I did ask each of our speakers to provide one fun fact. So at the age of 4, Angelique decided to be a bug doctor. And after achieving that, she decided to change fields and apply her old school neurophysiologic training to much larger species. So with that, please join me in welcoming Dr. Angelique Paulk.
Thank you so much for the excellent introduction, and thank you all for attending, as well as the program committee for inviting us and allowing us to actually present a lot of our work. What I'll be talking about is sort of capturing those tiny sparks of the human brain and using technological advances to be able to sort of address what's going on in the human brain in vivo. Because current microelectrode recordings have provided incredible insight into how the brain works, particularly in terms of brain-computer interfaces, as well as what's going on in terms of DBS and neuroprosthetics. But the latest technologies that have advanced the field, especially in the past 5 to 10 years, including neuropixels and thin film devices, have improved our spatial resolution, moving from 150 micron scale to closer to 20 or 50 micron contact-to-contact spacing. I'm going to zoom in on this lateral temporal lobe sort of view, and as you get closer and closer and get closer to the scale of actual single human neurons, which are illustrated here in blue and green, this is getting closer to that fundamental unit of the brain, and understanding that circuitry and understanding how those neurons interface would allow us to understand, not just cognition and basic function of the neuroscience, but also what's going on with pathology, such as in the case of tumors, epilepsy, and various other types of neuropathologies. Therefore, in this particular space, we have an opportunity that for every question that we ask of basic neuroscience, including cognition, we have an opportunity to address functional questions that could lead to clinical implications in the future. And in that space, we've actually been working in 3 different sort of levels of understanding human brain function. We work in the operating room, where we're getting an opportunity to work with patient participants who consent to ongoing research, and I'll be addressing that, as well as several of my colleagues will be addressing that specific space. Well, these are individuals who are undergoing already clinically mediated brain surgeries, such as removing tissue for epilepsy or tumors, to allow the patients to be either seizure-free or reduce tumor burden. In this space, we have a rare opportunity to approach the patients and ask if we can use these kinds of devices to record in tissue or on tissue that will be removed in the course of the surgery. The other 2 areas that I'm not necessarily going to talk about, but are very important aspects of or tools that we use to be able to understand the human brain, is in the semi-chronic space, where patients are recorded for up to 29 days with intractable epilepsy, in the case of intractable epilepsy, to understand where exactly the seizure onset is coming from, or in the chronic space, such as with the responsive neurostimulator device or deep brain stimulation. All of this is not possible without a huge and amazing team that we work with. So I wanted to first really highlight the fact that a lot of these devices, and bringing them into the operating room, bringing them through the regulatory hurdles that we've had to face, depends entirely on a huge team of engineers, neuroscientists, clinicians, and pretty much everyone along the way, all the way from the nurses in the room to the individuals who are actually designing and building the actual electrodes. And it's especially important to understand that working with this communicative team allows us to address and tackle a really tough thing to do, which is record from single human neurons. And in that space, I wanted to illustrate why we want to do this. Of course, I really am speaking to sort of like the crowd that would already know why, but in terms of understanding and capturing the scale of single neurons and identifying single cells in the human brain, it's been tough. And that basically a lot of the devices that are out there are often sort of single needles that are illustrated up here, that have a contact that has 400 microns in between contacts, which at the scale of human neurons, illustrated by these cells here, it's not necessarily going to capture what you want. So in terms of individual cell identity. In this regard, in this video, I tried to illustrate that when you're actually looking at ongoing activity and being able to identify single cells and single cell types, a action potential registered on a particular lone electrode, which is on the left here, is going to be different than if you actually have a high-resolution movie, so to speak, which is what NeuroPixels in particular has actually helped us reveal using these kinds of tools. This is in contrast to the wide array of the other kinds of devices we have out there. And I wanted to actually especially contrast this to the clinical devices that are out there. What you see are grids and depth electrodes up there that are at the same scale as what I'm showing in terms of the NeuroPixels probe or the Utah array. And it's a pretty massive difference. In that lower right corner, you can see actually a micro ECOG, which is a thin film electrode that has microelectrodes that are 30 micron space or sort of width, right next to and laying over a massive contact. And hopefully this movie will play, yes. But this will actually give you a sense of a scale with this particular image of when you're going from the whole brain all the way to that high resolution single neuron recording. You can capture a very different array if you go from a single electrode that's actually lowered and at lower resolution to what the NeuroPixels can afford in terms of like a spatial and temporal high resolution map of what's going on with the activity. It's for all these reasons and many more that we actually worked really hard to bring this into the space, both us and the group at UCSF, which you'll actually be hearing from Manny in a little bit. And to actually go from that lower resolution to high resolution, because in this second video I'm going to illustrate an ongoing recording, this is from the human brain, of activity. What you see along the middle is sort of the NeuroPixels probe, and the color coding is matching to single cells that are sort of firing along the depth of the probe. And so you have a spatial and temporal map of these single neurons that can be sort of identified across time and during a recording, especially in these acute recordings. Why is this important? Well, what we're hopefully going to be discussing, us and the rest of the group, is that cell identity actually matters, especially when you're starting to look at aspects of such as cognition, and maybe even a fundamental sort of human trait, which is language. In this space, the group at UCSF, as well as our group, have been, working very hard to understand what's going on in terms of fundamental aspects of language, such as phonemes, and how that's encoded by specific neurons in the lateral-temporal cortex and the dorsolateral prefrontal cortex. This opens up an entire different array of information, such as with a single recording, we can prospectively get about 100 units in that single recording and capture that much more information across different layers, across different cell types and possible cell types, and map that to ongoing awake behaving human being sort of talking or producing speech. The other aspect here, and this is getting back to this whole idea of actually getting higher resolution across the cortical layers, is a lot of the clinical devices out there don't actually capture what's going on at the different layers of the cortex. And we know massive amounts of computations actually happen at different layers in terms of input, processing, and output. In that space, previously all we had were these laminar electrodes, which are thumbtack electrodes used by a small set of groups that will allow you to look at some different features of oscillations. And we've used this to, as a way to validate our neuropixels recordings. So going from the laminar thumbtack electrodes and looking at the spectral sort of representation of oscillations across different layers, we actually see very similar types of patterns in terms of different frequencies being represented at different layers in the cortex, across multiple patients as well as individual patients. And then we compare that both with histology, with the thumbtack the laminar electrodes, as well as with three-dimensional reconstructions of the cortex using a number of different tools such as FreeSurfer to identify where we are, the angles and aspects of how it is the neuropixels are inserted into the cortex. The reason why that's important is that we want to then ask whether or not certain cell types are better represented in different layers, how they're activated in the different layers, and how they actually are involved in, say, seizure propagation, or on the flip side, in language or cognitive processing. Relative to this neuropathology aspect is for, as I had mentioned, for every single question that we want to ask in terms of basic neuroscience and cognition, we also want to be able to pay attention to pathology, looking at possible clinical implications. In past microelectro recordings, us and many other groups have actually identified what are possible neuropathology signatures, such as microseizures, which are extremely small but very large voltage events that happen in a very small amount of sort of microelectrode recordings that can actually be localized within a few hundred microns. We've used the Utah arrays and actually found this also with the thin film electrodes, which have 50 micron spacing, that allows us to ask, okay, when you look at these microelectrodes, do they actually tell us anything more on the clinical side? And in this illustration on the right, upper right, what I'm trying to indicate is that for a clinical lead in the same exact recording in the same brain, you can basically get one sort of like voltage trace that actually looks like what they call an interictal discharge, which is epileptiform activity. In contrast, that same area, that same space with high resolution activity, you can get localized high frequency oscillations and traveling interictal discharges and microseizures, which is substantially more information that has some type of pathological implication than just the clinical lead. And of course, linking that to the single neurons and single unit activity is absolutely going to be a major goal as well. An exciting space here, which also many of you have probably also sort of thought about as well, and has been a massive shift as of 2019, was basically this branching and massive expansion of cancer neuroscience or tumor neuroscience, a new field that links single neurons and what's going on with tumor infiltration and how it actually changes synapses between neurons through different types of molecular mechanisms. And we posit that basically these neuropixels probes and also thin film probes may allow us to capture and understand in vivo what's going on with these cells and tumor infiltration as well. Of course, a big piece of this was we had to make, sort of make a lot of regulatory steps and a lot of different steps to bring this into the operating room. And in this space, in particular, we actually wanted to share this approach in general, and we were sharing this. But Nature Protocols actually approached us to put together a paper that is essentially a protocol paper of how we actually. And what actually, brings a lot of pride to me is that this has been at least a useful tool to disseminate other groups to be able to sort of work with this. So that in fact, we helped Shraddha and people at Baylor to be able to start to do these kinds of recordings. And that, helped us sort of like communicate some of the practices we had for keeping devices sterile, for especially an important piece of us is, aspects of regulations and informed consent. And then, of course, different types of techniques of getting those recordings going. I will actually mention that human brain recordings, as in particular, offers a massive challenge. What you see here is sort of just a recording in the human brain. We're lowering the NeuroPixels probe, and you can see the brain is actually moving quite a bit. There is a lot of movement that doesn't happen with a mouse. In that regard, we worked with a group at Columbia who actually was able to, who also worked with a massive collaborative team to build a algorithm to be able to sort of do this kind of automatic motion tracking, and allows us to do these kinds of papers. So in addition to laying out and offering our techniques, we also are trying to offer and hopefully develop some better tools for analyzing the data. But I wanted to then go into the future developments in these last 2 minutes to mention that, yes, NeuroPixels is nice, but there might be even better tools, such as flexible devices that can actually be twisted and so forth, and so these actually can be inserted and move with the brain. And this is a case where we're able to do this in multiple species, in multiple form factors, and do this kind of testing for a thin film sort of micro SEG that can be inserted. Of course, I want to also especially advertise a talk that you'll be seeing later on, which is moving it even more into the flexible microelectrode, high-density, high-resolution devices, which Dr. Luan's going to present later on. And even more sci-fi in my perspective is actually this particular sort of approach was developed by Shraddha Shah and especially Youngbin Cho, who created a thin film electrode that actually has LEDs on the back of it, and those LEDs can light up based on neural activity that's happening underneath. So it actually kind of adds an amazing dimension of being able to see activity going on in the brain. We are very much, as part of this larger collaborative group, wanting to move forward, especially the Brain Initiative idea of open and sharing. So we've actively been sharing the human data, de-identified, of course, with the larger community, and that's actually been used in actually a couple of papers that I know of. And ultimately, these tools that I want to introduce, across these groups and so forth, may actually, answer questions that we want to tackle in terms of like on the fundamental single neuron level, with a huge variety of single cell physiological types that we even see just in the NeuroPixels recordings that we don't even see in the mice recordings. Our hope is that we can identify sort of like the underlying mechanisms for these different types of pathologies, especially in the case of tumors and tumor neuroscience, which is a brand new opening field. And translating these devices into the OR was challenging, but I think worth it. Thank you very much.
Thank you, Dr. Paulk, for that beautiful presentation. It is now my pleasure to introduce Dr. Manny Xu. Manny completed his PhD in neuroscience at Hopkins under the supervision of Daniel O'Connor, where he uncovered neuronal populations for sequence execution and organization using a novel sequence task in an animal model. Manny's currently a postdoc at the Chang Lab at UCSF, leading the direction of intraoperative NeuroPixels recordings in humans. He conceptualizes the fundamental aspects of human speech as a highly flexible and context-dependent process of sequence recognition and generation. Manny uses his background in systems neuroscience to approach the underlying mechanisms of speech processing at the level of single neuron activities and their circuit context. So, fun fact, Manny used to create science-related content on Bilibili and YouTube, and he hopes to continue soon. And considering that this is being live streamed, I'm pretty sure you can continue right now. So with that, please join me in welcoming Dr. Manny Xu.
Yeah, thank you, Lei. You know, like Lei really spent the time communicating with each of us to make sure like he know enough of us, and I really appreciate that effort. And I also want to thank the organizers and Samir and others for this like wonderful opportunity. Yeah, like.
You may want to advance a few more slides because I think it's still
Oh.
stuck on mine.
I see.
Forward.
Okay.
Or you can present the end of her talk.
There we go.
Good idea. Well, as folks have already seen from Angelique's slide, like 2 years ago, Angelique and colleagues from the Cash Lab or MGH group at large, and Jason Chun and Christian Sellers and colleagues from our group, the Chan Lab at UCSF, both demonstrated the feasibility of doing like NeuroPixels single unit recordings intraoperatively. And I was really like honored to be part of this effort, and that left me with a very unforgettable memories. So, and this really opens the door for to study a new dimension and with unprecedented precision and resolution about the neural circuit mechanisms of the human brain. Although like we are continuing like both groups with the technical development, and we are also gradually shifting the focus, as like Angelique also like cited the papers there, like to the ultimate goal of what we're doing here, which is understanding the fundamental questions. And for us, it's about the speech and language processing of the human brain. So here I'm glad to share our continued efforts to understand the circuit basis of a core mechanism. And that brings us to the title of my slides, the parallel linking of speech content from perception to production by single neurons in the human precentral sulcus. Gyrus. So even though speech perception and production can be often studied in separation, but we know they are interconnected, tightly interconnected aspects of the human speech. And here we can look at a simple block diagram from the top. We have the auditory input, and this input going through a cascade of transformations and eventually to the vocal output. And of course, the vocal output can act as feedback to the input, and things can be very complicated. But we love sometimes the block diagram because they're simple, but we also know that things are never as simple as the block diagrams. Just to make it a little bit complicated, but also not too complicated, to show like what's really challenging to understand the system is that the speech are also perceived and produced in sequences. On the top, so again, let's keep it simple. So let's say we hear a sentence, and then all we need to do is to repeat the exact same sentence. Simple enough, right? But let's see. I'm missing one more step here, which is like what we hear is often presented to the central nervous system as a spectrogram, and then it can transform into certain perceptual elements and hear phoneme, but not necessarily to be phonemes. And then something in the working memory. I'm just starting to making things up, and different elements, and they're not necessarily organized in the same sensor, but definitely related. And then to the motor planning aspects, and then the planning string them together and translated them into articulations. And here we see some like MRI images when a person is talking. It's already getting complicated, but more complicated is that these things need to be processed almost like in parallel. And while being processed in parallel, they still need to be internally connected. So then to get some insight into that, hopefully using the high resolution recordings, then we proceed to do the following task with the patient subjects. So this is a very simple task, a delayed sentence repetition task, where the subject first hear a beep and calling, hey, attention, now a sentence is coming. And then they hear a sentence, and for example, like nobody likes snakes. And then after that, there's a pause for a variable period of time during which they need to keep that sentence in mind and wait for another beep, and then they will say the exact sentence out. yeah, again, this is a very simple delayed sentence repetition task. And while the patient is doing the task, we get a chance to use the NeuroPixels probe Angelique has just introduced, and to record from in like single cortical columns and of the single neural responses. And here on the top right are labeled, I think, are labeled 20 recordings, and with yellow in middle precentral gyrus and red in ventral precentral gyrus, and the purple in inferior frontal gyrus, and specifically pars opercularis, and with the blue in the superior temporal gyrus of the human brain. But let's now first just zoom into one recording site, one column, and see what neurons are firing across the sentences. Like, just don't worry about the content, just like across all the sentences and across the different task phases. And we actually found in this precentral gyrus of the human brain, we actually saw perception activity and like activity during working memory and speech initiation and production. They actually tile the entire processing phases. And this is consistent with prior works, including works from our own lab using ECOG, showing that the precentral gyrus has very solid listening responses, but it remains mysterious of like what kind of like circuit computation is going on in there. Then just like zoom out a little bit, and then to show all the task responsive units that are recorded in the 4 regions I just mentioned. And so each row is a neuron, and I organize them in their preferred response phases. So with these activities together, and we can project them into a low dimensional space, just to see is there any like general phase patterns, and to aid our visualizations. So in this slide on the left, you will see such an embedding, like each dot represents a neuron, and the position of the dots is derived from their response patterns that we saw from the previous slides. And this is a video, I'm playing it, and as the subject is performing this task, and from perception to memory period to production, then we see the activity and the cells is brightening up from the lower left blue parts and clockwise, and to the delay period and to the initiation period in yellow, and then eventually to the production period. And on the right, showing 8 example neurons, and I sort of colored them by the best preferred speech processing phases. And of course, you also hear the clicking sound, which are like so endearing to the electrophysiologists, right? And that sound is from the trial of the unit 7, indicated by the arrow. So maybe we missed the sound while I was talking. Okay, so listen to this. Yeah, it's unfortunate. I am not being able to, I won't be able to play all the neurons in sound at the same time. Otherwise, we won't be hearing anything. But you can maybe imagine the sound of other neurons are making. And these example neurons are indicated by circles on the left. Yeah, I think that's at the end. I know the video, there are a lot of things going on in the video. It's hard to capture everything, and for the audience that didn't end up catching anything. So here is a static view showing like different neurons responding to specific phases at different sentences with very exquisite temporal structures. And we also have get the intuition from the video that the neurons are selective to content of the sentence. And here, just using 6 example neurons to show that, yeah, we can, even with a very low bar, we can like decode which sentence they're saying from their firing rate. Again, back to this important question. Yeah, we saw neuron in the precentral cortex that responding to both during perception and production, and we learned that many of them are encoding or selective to the content. then the question is, are they doing that independently during perception versus production, or there are some underlying deeper connections they are carrying out? So that's what we are trying to quantify, well, to find next. So interestingly, we found a neuron that fired like roughly at the same place during the same sentence, like Junior, what's the matter with you, between perception and production, and except that, and when we actually zoom in and align them, like dynamically align them phoneme by phoneme, we realize that activity during production precedes the activity during perception. So that fits the definition of a mirror neuron. So when I say this to a friend, say, hey, Manny, like mirror neuron is so out of fashion, then I think that's a good thing, because now if it's out of fashion, then we can finally just get rid of all the blows and then to focus back onto the original definitions and their responses and their circuit significance. So yeah, just to help visualizing this. But more interestingly, we found a new type of neuron that we call bridge neurons. So these neurons like started firing at this specific content during perception and continued the elevated firing until the same position during production, which is shown on the right. So these are just examples. So we decided to use a more unbiased method, a hypothesis-free method, to model them by different kind of transformation kernels. And you can see different kernels, hypothesized kernels, will capture the responses. And we fit that to all the neurons responding to both perception and production, and get those kernels. And we do a hierarchical clusterings across those kernels. And then we found interesting low intra-cluster distance clusters, and one fits the hypothesized mirror kernel transformation, mirror transformation, and one fits the bridge transformation. And here are the population overlay and the single examples. And here are just some more examples about like bridge neurons and mirror neurons. I think I'm running low on time, so I'm rushing through. So just to, so then the mirror neurons are not only neurons we saw activities happening at the similar points, and we also found these neurons. But when we zoom in and align the like individual phonemes dynamically, we don't see one activity preceding the other. We found activity are happening at the same time, and suggesting like these are purely auditory neurons, but they are special in the sense they are speaker invariant. So whether it's the same content heard from the speaker or it's said by the subject, even the tones and many features can be different, and they respond at the same points. And this provides the important substrate to offer a feedback. And we use the same method, and this is the same clustering, same modeling. And here I'm showing there's a third cluster that shows the feedback kernels. And here I want to just compare feedback and mirror side by side, because I know this can be confusing. And if you look at the overlay, and there's no difference between those in feedback, but we see that production activity precedes the perception activity during the mirroring. Again, back to this question, like how is the parallel processing and the system implement this parallel processing? And we found that in the precentral cortex, and different groups of neurons, specifically mirror neurons and bridge neurons, are doing this kind of linking that link information from one stage to another. So in the end, I want to thank my supervisor, Eddie, and for his like continued trust and support, and his like very vision to a very far horizon. And also I want to thank Queen Gracias, and who is the heart of our NeuroPixels team, like pumping the blood through the entire system, so like nobody else gets suffocated. So in the end, I have 20 seconds, 15. I want to share a quick fun fact. So at the beginning of this project, I created a GitHub repository and started to put my code, analysis code in it. And I named the repository np_babel NP stands for neuropixels, understandably, and Babel, because at this stage, I think the research is like we are seeing a newborns, maybe a twins, one delivered at Massachusetts General Hospital, another delivered at UCSF Hospital, and we are starting to see them babbling now, and it's amazing. And I think like I'm calling like all the panelists and the audience and the community to foster these twins together, or the kids together, and maybe 10 years later, 20 years later, and let's see what a eloquent person they become, signifying the pinnacle of human intelligence. Thank you.
Thank you, Manny. I could listen to the sound of single neurons firing all day long. Should be the intermission music for a crowd like this, just listening. And I have lots of questions, which actually prompts me to say out loud, we're really looking forward to the question and answer session, so please kind of register and remember those questions, and then we will have an opportunity, and there will be a mic for people to come up, both online and here in the room. So next, it's my privilege to introduce Dr. Shraddha Shah, who received her bachelor's degree in electronics engineering from the University of Mumbai. She received her PhD in neuroscience in August 22 from the University of Rochester, where she worked with Dr. Farron Briggs, investigating the neuronal mechanisms underlying visual attention in macaques. She's currently a postdoc with Samir Sheth at Baylor. She also worked closely with Ben Hayden and collaboratively with Andrew Watrous and Eleonora Bertoli, also at Baylor. Her postdoctoral work is focused on building a mechanistic and algorithmic understanding of brain circuits that are critical for efficient, flexible cognition, specifically in hippocampal and prefrontal circuits. Her research goal is to use multimodal approaches spanning circuits to behavior to develop a deeper understanding of how flexible cognition is disrupted in post-traumatic stress disorder, and to use this knowledge in concert with psychotherapy and neuromodulation to improve PTSD outcomes. Fun fact, Shraddha biked down the highest motorable road in India when she was, and I quote her, young, wild, and free. It's my privilege to introduce and please welcome Dr. Shraddha Shah.
All right. Hi, everyone. Thank you for that introduction. And I'm going to latch on to something that Manny mentioned in his talk. We have plenty of time to think about it. So I'm hoping that my talk will be somewhat of a pivot from what we've heard about, but also raise a lot of questions and really, hopefully, things we can talk about and think about for the next 20 years to understand the human brain. Sorry, I'm short. Okay, yeah, so I use this picture. This is like my desktop wallpaper, and I really like it because this is the murmuration. This is a starling formed from a murmuration of starlings, and I think we really, like, this is where we want to go in our understanding of the brain, like what are we actually seeing? And I feel these microelectrode technologies can give us access to these larger ensembles and kind of see what the patterns are. Okay, so for my talk, I'm going to focus on just reiterating why we are here, like what can we learn about the brain and what do we even mean by microcircuits, and kind of pivot into some of the work that I've been doing with Dr. Sheth and the group at Baylor in studying the hippocampus and recording from the human hippocampus. It's really early days, but I think there's a lot of like interesting directions we can take and think about. Oh. Yeah, okay. Yeah, and then talk about some of the results we're seeing and, again, future directions. So just as a reminder, I think most people are familiar with this idea of levels of analyses. And with the human work, as Angelique already eloquently showed us, a lot of our methods either give us access to the global activity, like LFPs or EEG if it's non-invasive, or we have access to depth electrodes, but again, much smaller yields. So we have some access to human single neurons, but not at the level of ensembles. And the critical piece missing between these bridges to build a mechanistic understanding are neural circuits, which devices like Neuropixels and what Dr. Luan would talk about can really fill that gap. And again, reiterating, zooming in, this means we can kind of describe the local architecture of the circuits. So we can look at different neuronal classes. We can identify what the different laminar compartments are. And especially for an area like hippocampus, where there's a lot of historical work in animal models in rodents and now monkeys, we can start building this data set across human patients to really understand what should we look for, because we don't really have a sense of what the ground truth should be for in vivo recordings. And we can, of course, look at neuronal tuning and look at in vivo connectivity patterns. So why hippocampus? I mean, hippocampus in human work and in otherwise, it's one of those areas that always lights up in the scanner, is always something that's active. It's like, all things cognition. And there's some beautiful rodent work, which again shows different layers and what the different patterns can look like. We have a nice motifs across different cell types. So most of you all have heard about place cells, grid cells, and you can look at the LFPs and think about sharp wave ripples, different kinds of theta oscillations, and some recent work has also shown how we can use different waveform patterns to identify cell types in the dentate gyrus. And so this is really amazing, and the stuff that I'm doing is not in awake patients, so all things cognition becomes a very kind of, debatable question. So what can we really learn from the hippocampus and humans in an anesthetized setting? So just as a reminder, the work that we are doing at Baylor, we are recording from patients who are undergoing surgeries for temporal lobe epilepsy, and we are doing a resection, well, not me, the neurosurgeon, and we can record from neuropixels. So these patients are not awake, and so we were wondering what can we really learn from this setting. And one of the things we know that hippocampus is classically involved in and important for is novelty detection. So on the left is a figure from a paper from 1996, where Dr. Robert Knight looked at patients with posterior hippocampus lesions and found that the novelty response, which is over here shown by a lack of sympathetic skin response, is completely obliterated in patients who have a lesion compared to controls, which is the solid line. And this was true for both auditory and somatosensory or tactile stimuli. And on the right is a paper again from Dr. Knight's group, more recently published in 2024, where they start looking at this in a more intracranial setting and find that you do find these classical signatures for novelty, which a lot of EEG people might know as P300. You can see that in hippocampus at the LFP level. So we thought, let's try this task in the OR setting when the patient is anesthetized and see if we find anything. So we use neuropixels. We're recording the hippocampus. We're still trying to figure out exactly which subregions, but just as a broader goal, we can see the hippocampus, and we are using an auditory oddball task paradigm. So auditory oddball task has been classically used across a lot of studies at different levels, which means it's a really nice task where we do know a lot about what we see electrophysiologically across different patient populations. And in our task, we had a counterbalance design. So we have two tones, where one tone is a standard and the other tone is an oddball. And for the first half of the task, one tone is a standard, and then we switched the standard oddball contingency, so it's counterbalanced. And so we can look at different things like tone encoding. We can see whether neurons care about one tone over the other, regardless of whether it was an oddball. Or we can look at things like whether neurons care about oddball encoding, regardless of what the tone was, because we have a counterbalance design here. Or we can see if we see any interaction effects, or what these days people call nonlinear and linear mixed selectivity. So that's kind of what I will show. But before jumping into the results, this is just an overview of our OR setup, which you all have already heard about in previous talks. And one of the biggest challenges for us as well was trying to find a balance between how can we reduce the motion artifacts and what would give us stable recording. So that was something that was a work in progress. But we use a combination of the ROSA robot, microdrive setup, and an awesome neurosurgeon to get hopefully good recordings. And as you can see, we can get both action, single unit activity and LFPs across 300 channels in the OR. And this is just an example of how high density recordings can be helpful to triangulate these units. And in my opinion, for an area like hippocampus, where it is known to have sparse firing because it cares about more specific things. This kind of technology can be helpful to also capture those units that may not be as high firing or may not be easily detectable. So I'm not going to spend too much time on this slide because we already kind of know these results from other studies. So it was a good confirmation that we also see ERP responses at, and the peak is at, the significant is at 300 milliseconds, which is like, again, the P300 response. And we're really curious to see what do we see in units. And we applied motion correction like Angelique discussed. And interestingly, we don't see a huge motion artifact, but it does make a difference in going through this pipeline. And on doing that, we find around 100 units, and with pretty stable waveforms, clean autocross correlograms, and as you can see on the right, the units are pretty stable across time. These are some example units with, again, stable waveforms. And what I think is really interesting is the population activity across time, where it's not the same, so you have a lot of variance, which again you would expect, but that's something more to look at. So again, looking at global activity for single units, if you're just looking at the grand average across the population of units, we're not really seeing anything clear. But when you start looking at single units, we start seeing differences. So to orient you to the graphs, in blue is response for oddball, in red is response for standard. So in the top, you can see that this unit does show a differential response to oddball. And then in the second panel, we can see that this neuron shows a differential response to which tone was played, regardless of whether it was standard or oddball. And then I'm not going to explain the last one because I can show you over here that when you model the activity of each neuron to look at tone encoding or oddball encoding or an interaction effect, the most dominant population is neurons which encode tone and oddball interaction effects. And the next most dominant is the oddball effect. And what I thought was also really interesting is the surprise effect or the change effect. So if the neurons only cared about whether the tone changed from the previous tone, that would be the dominant population. But we don't actually see a lot of units which do surprise encoding or change detection. So it's truly caring for some sequence encoding, which again, we know about hippocampus, but it was really interesting to see this in anesthetized patients. And this is to drive home the point that when you look at the distribution of the units across the population, if you focus on the orange pie of the pie chart, or orange piece of the pie chart, that's the most dominant population, which shows the tone oddball interaction. And these are like what people call canonically the conjunctive encoding or the nonlinear mixed selective units. And if we look at just units that care only for tone, that's the very tiny pink portion. So most units in the human hippocampus show some version of oddball encoding, either linearly mixed with tone encoding or independent of that, or non-linearly mixed with tone encoding. And we can then start also zooming out. So we have these amazing simultaneously recorded neuronal ensembles, and we can start thinking about these amazing methods that people have developed over the years to look at population coding patterns. And one thing that we just tried is looking at the regression weights that we have from the previous results. And if you correlate the regression values for tone encoding and oddball encoding, you can start asking questions like, what is the format of the population coding? So over here, we can see that when you correlate that, there is no significant correlation, which suggests that both tone and oddball are encoded in an orthogonal format. So for downstream areas trying to decode that, the codes are not, the codes are independent, which makes it easier. And then similarly, if you correlate the absolute values of the beta coefficients to see whether similar populations were upregulated or downregulated, we can again find that there's a small, like a significant proportion of overlapping populations with independent codes. So this starts kind of giving us a sense of what the population is doing and can maybe help us address some questions like questions about pattern separation and completion and which brain region is involved and how hippocampus is solving that issue, something that we can't easily address with questions where we don't have enough units. So zooming out, we can do a lot of fancy methods that I'm hoping to do soon. But yeah, like correlated variability, which is important to understand information coding properties of units. And we can do state space analyses. And this example, I think, is really interesting, where they look at population entropy measures across mice with Alzheimer's and without Alzheimer's. And you find that there is a difference in the entropy, suggesting that with Alzheimer's, the amount of independent patterns you encode is much smaller, which is interesting from a mechanistic perspective and gives us a new insight into the pathology, which previously would have been impossible. And these are things we can do in humans. So ongoing work is kind of looking at cleaning up the data, as Angelique probably already convinced you all that this is complicated data and there's a lot of work that needs to be done to get to a point where you're confident. We are trying to develop some electrode localization methods with Dr. Sarah Hellbronner. It's a very difficult but important thing, especially for hippocampus, where we do know that different subregions are really specialized and we would want to get at that. And a lot of different unexplored directions, but I think with this you can do like more patient-specific stimuli. And I personally am also very interested in looking at questions across like development and patient populations, and what do different electrophysiological patterns look like across these different populations. And again, a lot of questions, especially like Dr. Buzaki has talked about, looking at spontaneous activity in hippocampus. So like Angelique said, a lot more work than one person or one group can do, and hopefully we have plenty of time to think about it before we get to work. But I would really like to thank Dr. Shed for trusting me with this project, and Dr. Ben Hayden, Andrew Watrous, and everybody else in the team, and especially Angelique. And I have a fun fact with that as well. When her paper came out, I was still in grad school, and I remember seeing that paper, and I was like, this would be really cool to do in humans, and never thought that in my postdoc I would be doing that. So yeah, thank you, and thank you for your time and attention.
Thank you, Shraddha. It's just truly amazing, and so many questions that I will again suppress to keep us going, but hopefully we'll get a chance. The last but certainly not least, it's my pleasure to introduce Dr. Lan Luan, who was trained as a material and quantum physicist, received her PhD from Stanford and then postdoctoral training at Harvard. She then transitioned over to the good side in neuroengineering, and joined the faculty at Rice in 2019, where she is associate professor in the Department of Electrical and Computer Engineering and the Department of Bioengineering, and is also a core faculty member of the Neuroengineering Initiative at Rice. Her research focuses on the development of large-scale, bidirectional, flexible electrodes for high-bandwidth, long-lasting neural interfacing. She also integrates electrophysiology with optical methods and investigates the neural underpinning of novel neural prostheses and major neurological disorders. The overall goal of her research is to provide technological and scientific advances to restore impaired neural functions. Fun fact, Lan's last and first names are very similar. This was deliberately designed by her dad because her first name is common mispronunciation of her last name in Chinese, and so this way people are always correct no matter which way they pronounce it. So with some personal hope that I get it right, please join me in welcoming Dr. Lan Luan.
Thank you very much for the very nice introduction, and thank you for the organizers to have me presenting here. It's a great honor. I will switch the gear a little bit and talk about the engineering side, about our work of developing ultra-flexible, bidirectional, and long-lasting interface with the nervous system. First, I have a financial disclosure. I'm a co-founder and hold equity of Neurothreading. This entity currently is commercializing this flexible electrode technology with the focus of better serving our scientific community in capacities like academic lab cannot do. So recording and stimulation technologies are of critical importance for us to understand and treat disorders in the nervous system. Largely fueled by the Brain Initiative, there have been booming development of high-definition electrodes for animal research. Here I really show a partial, incomplete list of representative work in the past decade, ranging from silicon electrodes, neuropixels, and microwires, and carbon fibers, and flexible electrodes of various kind. So those electrodes made it possible in many, many labs across the world to record hundreds and sometimes thousands of neurons in one animal brain possible. And that has really revolutionized how neuroscience is conceived and performed. In stark contrast, when we look at human applications, the options are still very limited. Most of the tools we have rely on these microscopic electrodes, such as DBS and SSG electrodes that only have 4 to 16 contacts. And even in a more novel design where microwire bundles were threaded and then protrude out of SEEG electrode to fan out to record single unit activity, we're still looking at recordings of a handful of units. Utah array have been implanted in human patients for about 2 decades, but so far has been about only 30 patients receive those implants. We heard this amazing works from the panelists about neuropixels recordings in human. However, I think we all recognize how scarcely this can be done these days, and they're still limited in intraoperative settings. So Angelique gave an awesome technical overview of all the electrode technology. Adding on that, I just want to re-emphasize the size difference. If the entire screen is the SSG electrode, the neuron size is actually this tiny dot right in the middle of the screen. We need electrodes to be we need human applicable electrode to be much smaller to approach the size of neurons to reduce invasiveness and also to improve resolution. So we heard all this amazing work of neuropixels in intraoperative recordings, but we also heard over and over again the concept and phrase of motion artifacts or motion corrections. And this is thought to be because of the mismatch mechanical mismatch between the hard electrode made of silicon and also the very soft brain. So because of this mechanical mismatch, as the brain moves with heartbeat, respiration, and other movement, the electrode cannot move with it. So that leads to the motion artifact that all these authors and researchers spend extra effort to take care of. There is one other issue of rigid electrode that has not yet shown up in the intraoperative recording, but very well known in the animal research world. That is the adverse tissue response over chronic implantation. So if you leave those rigid electrode implanted in the brain for weeks, then the tissue will react adversely to it, signified as this glial scar encapsulation and neurodegeneration. And as the result, the neurons nearby are degenerated, and your signal of single units are really fading out. So motivated to overcome these challenges in rigid electrodes, over the past decades, I developed flexible electrodes together with my colleague Chong Xie, and we named this nanoelectronic thread, or NETs. I will just refer the short form NETs for the rest of my talk. So NETs are made on polyimide. They have this very regular layout of electrodes, as you can see, and also over the time and generations, you can see our emphasis on scalability. We pack more and more electrode, and the newest generation have hundreds of them in one tiny thread. So there is one key feature of nets, that is its thickness is only about 1 micron. Within this 1 micron thickness, we managed to pack in multiple layers of insulation, interconnect individual electrode contacts to record high signal neural activity. And it's this 1 micron thickness that gives us the ultra flexibility you see. All of this photos were taken in water because they do not freestand in air, and you can see how easily to deform them. So our design principle that drove to this extreme form factor of just 1 micron thickness is to minimize the interfacial force, force between the electrode and the tissue. And when we examine the interfacial force, what matters is bending stiffness, the force required to bend the electrode rather than how flexible or soft the material of the electrode is. And as shown here, the bending stiffness scales with the thickness T to the 3rd power. This means if we thin down a structure by a factor of 10, we gain 1,000 times in the bending stiffness. This is intuitive. I always give this example. Sorry if you have heard this before. Like the tabletop and the piece of paper are both made of wood. The paper is so floppy not because of the material, just because it's much thinner than the tabletop. So this is exactly what we did by thinning down the polymer electrode from tens of microns to only 1 micron. You can see on those pictures like how much more flexibility we gain. And this is and when we quantitatively measure it, the force we need to deflect those electrode is only nanonewton. That is in the range of cellular traction force. We believe this tiny force is critical for the stability of the recording we have in those electrodes that I will show in the next several minutes. So the electrode is so flexible, it does not freestand in air. We have to send it implanted in the tissue. And we use shuttle devices to implant them with the 3 design principles. We need to have minimum footprint of implantation because any trauma larger than 100 microns will be irreversible. We need precision because we need to have fine control of where we send those electrodes. And finally, we want stability because we want to send hundreds and thousands of them into the brain. And we end up having maybe 2 different strategies. One use a sewing mechanism where the electrodes goes in hooked up with a tiny hole of the shuttle device, just like sewing thread into the brain. And another is temporary adhesion. But in both cases, we implant the shuttle device and a net bundle into the brain and then retract the shuttle, leaving net implanted where we want it. So how does the tissue probe interface look? Here I show in vivo 2-photon imaging of the interface because back then I really want to see how the tissue probe interface look. For that we have a special surgical preparation where we co-implant a cranial window with the electrode. This way we could show you the tissue probe interface of a very thick stack of 500 microns, but also could monitor the longitudinal evolution of the tissue probe interface. Here, what you see is a thick stack of vasculature in vivo 2 months after implantation form a normal, healthy vasculature around the probe, which is red. And also when we came down to measure the microcirculation on that microcapillary right by the probe, sorry, I do not have a laser pointer, we have the same level of microcirculation with the probe even touching the capillary. I want to clarify that implantation does make an initial trauma, but because NET is extremely flexible, the initial surgical damage get repaired over time in about 2 months. Here I show you vertical sections from in vivo 2-photon imaging, and you can really see the regeneration and reperfusion of vasculatures near the implantation. And at the same time, the recording, we could record more spikes and more stable spikes as well. Here is a glimpse of a recording from that. You can see, and we often could record months after implantation and sometimes to the end of the animal lifetime. You see we could capture exactly the same set of features of the waveform over days and months, but you also see these changes in the amplitude from this recording, and when we record more sparsely, the changes is larger. And this is consistent with two-photon imaging of exactly the same location over 4 weeks. What we see is general stability of the tissue of the neurons relative to net, which is red in those images. But in some occasions, we do see very slow, small migrations of neurons. And because the migration is very slow, if we record 24/7 for 30 days, and you can see we really faithfully track the same neuron and also able to capture the subtle changes in the amplitude. So this is a scalable approach. We could do this, basically stack many of them together, and we show the thousands of channels in one mouse brain. Because they are only 1 micron thick, they are actually very small in volume. This allows us to pack them very densely volumetrically. Here we have over 1,000 contacts in a volume of 1.3 millimeter cubed in a mouse brain, and this allows us to examine the connectivity in the microcircuit, and here I show those connectivity changes in response to visual stimuli. To verify that this flexible electrode could record in highly mobile tissues, we implant in this lumbar spinal cord of a mouse. And in the video, you see the synchronized behavior and recording. And just to convince you, we can hold on to individual neurons regardless of what the animal does, move, stop, initiation, motion, whatever. We spent brutal force to make every contact on that bidirectional. That means they can robustly inject a current up to 50 microamps, and we've tested 50 million pulses in vivo. And our in vitro, and our in vivo measurements, here I show a behavior task where the mouse is trained to report the detection of stimulation through a go, no-go wheel turning task. And you can see across all 5 animals, the behavior data as a function, the threshold as a function are shown here. There are 2 features of this. First is how stable the behavior threshold has been. If you look at the bottom curve, you will see over hundreds of days, the detection threshold did not even change by 1 microamp, which is the resolution of our stimulator. And another feature is just how low those current we needed. Right here, we only need 1.5 microamps or 0.3 nanocoulomb per phase to maintain the behavior detection. If I put this threshold together with all the chronic implants in animal models, you can see the star is our result. You can see where orders of magnitude lower than the other studies. So this support our hypothesis that improving the tissue-probe interface could also improve the stimulation fidelity. The single argument is just the effect of distance. Without scar, the stimulation site is much closer to the neurons, so we can use a much smaller current to elicit a focal effect. This provides higher resolution stimulation, safer to the tissue, and also reduce the chance of device degradation. And our post-hoc histology studies verified this. You can see both for the passive and the stimulating side, we did not elicit tissue response. Importantly, all the stimulating contacts could record high signal to noise action potentials for the entire duration of implantation. There are also a couple of animals we forgot to euthanize, and we record till week 76 after behavior training. So finally, I just want to say this is not, with the general support of Brain Initiative, we're able to disseminate the tool to the broader community. And this has been in the US, in Europe, more than 17 labs now. And we have been optimizing this for diverse animal models, both for the brain and for the spinal cord. Finally, I just want to show my wish list of transformative neuroelectrode for both animal and clinical application. I think we are seeing the light for animal research. It's a longer path for human application to realize all of these goals, but we'll all strive to make those happen. With that, let me thank my group and the general support of NIH. Thank you.
Really beautiful work. We're clearly seeing the future. It's amazing. Let me invite anybody who'd like to please come up to the microphone and just form a little queue there to ask any questions. Also, there may be questions online. I don't know whether it's the chat or the Q&A box, but please do enter them there, and please.
Hi, really nice symposium.
I'll ask you to break for 1 second. I don't know if there's a button on that mic or whether we need to do something in the back.
I don't see a button. But
You're good. Here.
Okay, so I have 4 quick questions for Manny Xu. I really liked your talk. I thought it was pretty exciting. One thing you didn't clarify is whether or not there was a difference in the presence of the bridge and the mirror neurons in depth. Did you do an analysis of that, and would you expect to have, say, more activity in the bridge neurons in lower layers? Should I go with the other questions now, or you want to answer this one at a time?
Let's do one at a time. I have a short working memory. So due to a limit of time, and of course this is just one part of our findings. And yes, and we looked at the depth, and we are seeing some trends of unequal distributions between mirror and bridge. But we are still working on resolving the details. So maybe.
Okay, so
they'll be answered in the future.
Okay, so in your paradigm, the subject repeats a sentence that they've heard already. Yes. So I wonder whether or not you would get different responses for the bridge neurons if they repeated it, say, in the opposite direction, or they gave a different answer. So for example, if you gave them the numbers 1, 2, 3, and they repeated 3, 2, 1, in other words, are they encoding the sequence as presented, or are they initiating a response and holding a response? Have you tried different sort of behavioral paradigm?
Yeah, short answer is we haven't tried other variations yet.
Okay, and is it speech related, or if you played a sequence of musical notes, would you expect to find the same set of cells? Or is this really related to language?
From our data, so again, we haven't tried other variations, but from our data, we do have the auditory cues, like before the perception and before the production, which is one calls for attention, one is a go cue, and we haven't found any quote unquote neurons mirroring the cues, or I don't know what that the mirroring is going to be, or bridging from one cue to the other, and then the 2 cues are at different frequencies. So I think that's the closest I can provide for your answer, but we haven't done exact tasks you were doing.
Okay, quick final one. Do you ever present visual stimuli instead of a spoken stimulus? Would you expect to get, say, motor cortex to respond to that, even if they had to produce the sound? There might be subvocalization when they see the visual input.
Could be. I haven't done it in my project, but in the literature, middle percentile has been suggested to mediate aspects of reading, and those evidence were from not from single unit data, but that's what we learned from the literature. So I won't be surprised if there could be some process involved. Yeah.
Okay. Well, thank you.
Yeah.
Thank you very much.
Thank you.
Thank you. I think we have a question from the net. Is that right?
Yes, hello. There's quite a few people tuning in online, so thank you for the wonderful session. I'm just going to ask 2 of the questions so far that are on there. The first is, I think, directed towards Dr. Luan. Have there been any advances in inserting thread electrodes? Although the electrodes are small, the insertion damage with a 100 micrometer needle in high density would be significant.
Yes, okay. Yes, that's a great question. Yes, the damage is dominated by the size of the shuttle devices. And for that particular reason, we actually try to use as small shuttle devices as possible. So the size of our shuttle devices range from 15 to 50 microns for rodent experiments. We never go to 100 for rodents. Yeah.
Great, thank you so much. And then another question that I think is directed towards the entire panel, I think it's more of a hypothetical. There are some people in the chat online who are MRI technologists, and they want to know how can they incorporate some of this high-resolution data or high-resolution models to improve lower-resolution technologies like MRI, like low-field MRI, if you have any thoughts on that.
Thank you. I don't know if we can, I mean, we are trying. And in fact, there's, in terms of the clinical workflow of patients that are actually being implanted with electrodes, they actually undergo fMRI and various other types of scanning procedures, sometimes every so often the 7T, when depending on either the case or actually some aspects of them actually being willing to participate in research. And it's a very much of an active field of being able to then take that, say, preoperative image, match it up to the 3 dimensionals of mapping of what happens in the intraoperative space, which in this case, we're actually trying to, image match where exactly we put the electrode relative to the brain regions. And then ask, is that going to be sort of pair up with, say, layer specificity in the fMRI space and what we see for the neurophysiology. So actually, to answer your question, that's what we're trying to do. But that's what we are trying to help inform that kind of level of specificity. And that's why also I had highlighted that layer specificity and depth specificity is absolutely critical to answer and maybe allow us to have these different modalities intersect as much as possible.
And maybe I'll just add to the imagers, there is this opportunity to be doing human HCP mapping, to be using human cortical parcellation mapping before potentially some of these clinical procedures. That's, of course, a research event that would happen. But I was struck by listening to some of the presentations that we have a group of true neurophysiologists, single unit neuronal ensemble neurophysiologists, and the labels on your slides said precentral gyrus. They didn't say area X, which I think is an evolution, because the same presentations 20, 25 years ago might have said that the bump in front of the central sulcus is the primary motor cortex in a person, and then maybe 10 years ago, it might have said something else. Maybe it would have said area 4, and then maybe a little more recently, I'll be off by some years, maybe would have actually said area 6. And as we know to that question, or as we think we know, at least histologically, the Brodmann 4 ends at the anterior bank of the central sulcus and slowly climbs up, and then we find our way in humans, at least to area 6. So I'm hoping, in answer to the question from the MR imager on the net, that we'll actually begin to ask these very agnostic questions of, okay, we have a human parcellation map that says that we should be in functional area X, which is based on some really good high-resolution imaging data, and do we have neurophysiology, the additional single unit neuronal ensemble physiology to back that up? And if we do, what do we learn, and what are, at this resolution, the differences between those two? For example, even in just this kind of precentral gyrus between area 4 and area 6 in any functionally interesting manner. So I may ask you more about that, but more importantly. Tim.
I have a question for Lan. We have quite good success in mice with neuropixels. It's not unusual to get 6 or 9 months of really good stability in a mouse, even as posterior as visual cortex. I'm wondering, do you have data in larger animals like rats where posterior becomes a major problem? And I would like to see long-term stability in cerebellum. What have you got so far?
So right now, our rat testing is performed from tester labs. We do not yet have all the data organized, but we will, we're looking at the cortical areas and also hippocampus. And once the data is organized, we're going to share with the community. We also had a couple acute recordings in domestic pig models where we could hold on to unit during surgery without seeing large amount of motion artifacts. So those are the data we have.
Hi there. Very nice talks, all of you. Thank you for those. My question's for the entire panel. I was hoping you could speak to some of the challenges I imagine you would run into just handling the sheer volume of data you're getting from recording from these high-density things. So I was hoping you could kind of talk about that a little bit and things that you're doing to mitigate those challenges.
It is a lot of data. It is a lot of data that there are actually some great tools out there for this, but a lot of the data really isn't compressible in an easy way. Really, we're trying to find ways to sort of make this sort of like both data shareable, usable in a larger sort of community, such as like converting into common formats such as NWB or in the Dandy format. But also, we're going to start to have to rely on the cloud, sort of like storage process, state locations, because storing it locally is, I think 10 grand a year, or at least 10 grand a month. And so it is actually a major challenge that we've also relied on DABI, which is one of the Brain Initiative funded sort of NIH sort of data repositories. But that doesn't necessarily, it's not really feasible if you want to do local data processing with your data, and have a hot sort of like storage, so to speak. So we are kind of working with a very cost-heavy sort of like burden, and we don't have a great solution, honestly. And ideas on this and directing funding toward this would be fantastic, because we are, if we want to analyze our data and still have access to the terabytes of data that we are gathering, especially for the human data, we need to have it on a protected server. We need to have it encrypted. We need to have it all sort of protected such that we don't have like patient identifiers sort of shared out there, et cetera, if we need to actually have it linked to a clinical indication. So we don't have, we have a way that we're doing it that is expensive, and I wish we could figure out another way of doing it.
Thank you so much.
No, all right, that's our storage answer from the panel? Okay, that's our storage answer. So if there are additional questions, please come up. Shraddha, if you will, doing this amazing high-resolution research, and I know you have an interest in PTSD. Can you connect these for me? How are we going to take the tools that you're developing and the research that you're doing and bring that to this incredibly complex and important disorder?
Yeah, no, that's a great question. Okay. Yeah, no, that's a great question, one that I ask myself many times a day. And yeah, I was a bit skeptical if I should put it out there. So I think one of the things that circuits can help us, and this is maybe not directly answering the question of like what I will do, but I think for the field indirectly is through incorporating theoretical models. I feel that this could start providing us a ground truth for what models of the brain could look like. And I think building on top of that could give us understanding of what at a more cellular level is disrupted across different patient populations. I think there's a lot of unknowns, and I alluded to like the developmental aspect that I know nothing about, but I've been like really fascinated with that since I started grad school. And the first time I took a systems neuroscience class and I was forced to listen to someone talk about developmental neuroscience, and was really fascinated that there are these really amazing things. And then we all know with some of these brain areas, they develop through our maturity. And I think just understanding what these different signatures could look like, where I have honestly no idea what we would find if this is the right technique to do that. But I feel the only way to figure out is by trying. So that's kind of where I'm getting at in terms of the circuits. And I'm hoping to branch out. I mean, I'm, using the things I was trained to do, which is more like connecting behavior to neural activity. So yeah, I think doing more with that. But it's not a straightforward connection, and I have a lot of thoughts on like PTSD research in general, which don't directly connect to what I'm doing right now. But I think there's a lot of scope in really characterizing more cellularly and think about how this relates to DBS and stuff, which I think a lot of other people are doing.
Thank you. Yes, please.
More questions from online. For Manny, if you ask a subject to suppress a word as part of your paradigm, does that change the neural activity that you see?
So I guess the short answer, again, apologize, is like we haven't tried a lot of variation because intraoperable recording opportunities are so rare, and we have already at the limit of collecting the data we need to have. And also like suppressing, meaning like instruct the subject like not saying a certain word. That could be a very interesting manipulation.
Thank you. And then this one is, again, for the whole panel, kind of looking forward. Is it viable or feasible, do you think, to use neuropixels or similar technology to inform better brain-computer interfaces or even incorporate them into better brain-computer interfaces? And maybe this goes back to the difficulty with managing the amount of data that they produce. So love to hear your thoughts on that.
I have one thought on that, going back to one of the things I mentioned later in the talk about zooming out and looking at neuronal populations. So there's some really cool stuff at, I think, CMU from Byron U, where they look at population manifolds and looking at how that changes with learning, and that research is geared towards like BCI development. And I think these technologies, now that we can look at neuronal population coding and changes in that with, I mean, learning is something that you could do if you have an awake patient, but even in this setting, we can start kind of thinking about questions or even the activity in that direction, which was previously not possible. And I think it at least gives us a bridge to some really cool work that they're doing in the motor system. So I think a lot of directions, which I feel we need to be connecting, like, it depends on what brain area you're recording from, but I think that could be something really interesting.
Just wanted to speak to that, and more or less, like, what's great about sort of seeing all these talks together is also, in addition to what Shraddha showed, also what Manny sort of like amazing results there, is that the cell identity and cell specificity that these kinds of devices afford, being able to identify these single cells and maybe even get at cell types, that's a big difference, no matter what. And that kind of specificity may be absolutely key for sort of untangling this really complex issue of we're recording extracellularly. We're not inside cells. We're not imaging the cells. We can't do that in the human brain. We can't actually, just sort of, tag a cell right now, and you look at it with optogenetics. But we're listening in the dark, and in that space, we can actually capture that kind of information that much better with higher resolution using neuropixels or these high resolution devices. And that's why we're so excited as electrophysiologists. And every time I see a spike, I'm then imagining those sets of cells in the background. So, for brain-computer interfaces, my hope, and this is just a wild idea, but my hope is that basically the cell identity specificity may improve those brain-computer interfaces, may improve those devices. And I know that we know that IMEC and various other companies are very much engaged, and that is the big main goal in the end. Of course, we're chasing. There's aspects of Neuralink in the background, of course, and everything like that. But there's also the fact that many of these companies want to go after this. So, and I do think the higher resolution is going to keep on improving how well these brain-computer interfaces work.
So from technical development perspective, of course, flexible, large capacity brain-machine interface is what we strive for. I think in addition to the cell specificity, high fidelity in single unit recording, several additional benefits are like maybe we could have access to deeper brain regions, no longer limited in the superficial cortical regions. And also, if we have the flexibility of designing electrode, we could choose how to distribute the context, either through one thread or through multiple in the region, and this will be tied to the specific application and the brain region of the brain-machine interface. So I do think this is a goal, one of the goals for technical development for sure.
And I think with this last great question, what this really speaks to, what the panel addressed today, is that the BRAIN Initiative has just been so amazing in not only helping to develop the tools and the analysis techniques, but also the clinical research that has de-risked this field for now multiple industry partners who are joining, who are taking the original technologies and moving them forward, thinking about clinical trials, and really being able to bring this incredible fundamental neuroscience and human neuroscience that we're learning in the OR and nearby through all the way to developing new therapies for these complex neurological and mental health disorders, which I know is really exciting for all of us in the room. And with that, I'd just like to thank everybody for joining us for today's symposium, and one last thank you to our incredible 4 speakers today.